whatever, please give it. So who's supposed to pick it up? So they can contact Shaptoshi and Nandini. Yeah. So Nandini is outside, so we will introduce her when she's in. Shaptoshi, can you just stand, wave your hand or something? Yeah. So you could just hand it to Shaptoshi and Nandini. And secondly, many people have been asking us about Wi-Fi. So Wi-Fi is actually very simple. When you connect to IMSC Wi-Fi, login prompt will come up. And so apart from the username password, below that you will see like a link saying create your own account. So just create your own account with your own username and password. Just make sure that the username is sufficiently different that it doesn't clash with any of our existing usernames. So you know, if you accidentally happen to have chosen a username which is already there, it will say that another username of that already exists. So then you'll have to choose another username. But most important of all, you have to give a IMSC collaborator name and email ID. So you can give my name as the collaborator name and my official email ID, that is shitabro@imsc.res.in, as your collaborator email ID. And then it will ask you to validate. So you have to then, you will get half an hour of Wi-Fi. So within that time, you'll have to go to the contact email of yours that you had given, and you have to click on that. So then it will validate your account. Okay. So that's the process of creating our own Wi-Fi. If you have any questions after this session, you can again catch any of the volunteers or catch me or Shakti, and we'll explain it again. Another point is that between the end of the session and dinner, which starts about 7:30 at the other end, at the across the street in the cafeteria where many of you hopefully had breakfast today. So between that time, if you wish to discuss with your friends and group members, you can use the 4th floor coffee lounge, which today, if you'd like to know where it is, again you can catch any of us. We will show you where it is. So between the end of the program of the day and dinner, you could just hang out there and use your laptop, discuss with your friends about the projects and so on. And finally, our library is open during working hours, which is I think from 9:30 till about 5:30 or 6. So if you'd like to consult any books or something, you can just visit the library during that time. Okay, so with those announcements, let me now give the floor over to the first speaker of the program, Professor L.S. Sashidhara. So Sashidhara, of course, needs no introduction, but I'm fortunate to have known him for a long time now, since when he was in the Center for Cellular and Molecular Biology at Hyderabad. So we have had the fortune of on multiple occasions to invite him over to give a talk, and actually my interest in developmental biology was spurred by Sashi's talks on the complex system school that we had here back in I think 2010. So Sashi gave a wonderful set of lectures on developmental biology, and that's when I first got interested in this. So he's been my kind of teacher in that respect. And eventually Sashi moved to first ISO Pune, then to Ashoka, and finally now he's at NCBS. So without further ado, let me invite Sashi to give the inaugural lecture of the workshop.
Thank you. Thanks, Shitabro, for the invitation. It's a pleasure to be here. Also, accommodating my change of schedule multiple times. And apologies for that. Today I'm going to talk mostly about what is sort of known over the last 60-70 years in the area of developmental biology. And the next one, perhaps I'll sort of pose some new questions. To start with, a disclaimer: I'm not a computational biologist, not a quantitative biologist, not done any modeling, although we have collaborated with some people. My area of work is experimental biology, using experimental approach, using modern techniques of cell and molecular biology. How to understand development is what I'm sort of pursuing for the last 30 plus years now. And only recently sort of started some collaboration with mathematical modeler Mandar Inamdar at IIT Bombay, with on role of some of these mechanical forces in promoting the shape and size of organs. I just want to first understand the audience here. How many of you have had some biology background among the participants? Oh, that's quite a few. At what level? Undergraduate level? Okay, not bad. But I assume that you've not done biology beyond 12th. Doesn't matter even if you know more than that. But I'll just, for the flow of my thoughts, I think it'll be easier if I start from the scratch. I mean, again, for this audience, we need to start from the introduction that every time an organism is born, it's born as a living organism to start with. Second is born with all the morphological features and, of course, all the internal features of a specific species it belongs to, right? It could be a human, it could be a plant or any other animal. And the third is it inherits certain specific traits related to the parents or slightly a larger group which are connected genetically to that particular group, what you call as family traits, right? It's not working well? Oh, I think it's working fine there. Sometime. I didn't realize. Because I saw that it's working well. Should work. What's it? Maybe I'll start from here. It just worked some time ago, right? Yeah. Okay, sorry for that. No, it's okay. I'll just stand here. So until we knew something about how these traits are inherited, right? Everything was a black box. Life itself was a black box, and the development, how an individual is born, true to the species type, with lots of features related to the family, everything was a black box. It was also a black box in the context of biodiversity, right? There's so much of variation. There is so much of variation within the families, within the population, between populations and species, and then across the globe, right? So given all of these things, the major breakthrough happened when Mendel sort of studied the patterns of inheritance, right? And he showed there is a specific pattern in which these characters are inherited. And with a lot of quantitative, he's the first quantitative biologist in a way, the way he did his experiments. And subsequently, about 50 years later of Mendel, part of his work was, much of his work was unknown to the scientific community, whatever the small scientific community we had at that time, mostly in Europe. It was not known to other people who are working in the area of cell biology or developmental biology or taxonomy and so forth. And in parallel, Darwin was working on evolution, and he proposed his theory of evolution, and there he talked about there's some mechanism by which the population will have some variations, and these variations, when they're subjected to environment, and then certain variants reproduce at a higher frequency, some at a lower frequency, and that leads to changes in the population structure, and then overall new population features. And cumulative effect of all of these things will lead to new species or new forms, right? That's what Darwin was talking about. And sometime at the end of 19th century or beginning of 20th century, when people rediscovered Mendelian laws, And they found out that it is true for across all species that is studied at that time, whether plants or animals. Mendel himself studied using pea plants as model organisms. And then people found out with connecting to Darwin's theory of evolution to genetics, and when the two were connected, and then they decided that perhaps we should all work on one or two model organisms, and whatever we generate that knowledge on any specific model organism will be applicable across different species because of evolutionary relatedness of all organisms. And in fact, Darwin himself had proposed at some point in his book Origin of Species, the last chapter, that paragraph, he mentions that perhaps there was a single origin of life, right? And given that at some point they're all related, and we should be able to extrapolate to other organisms, and in search of model organisms, which were found out to be this Drosophila melanogaster, fruit fly, found out to be very useful, which was used by Thomas Hunt Morgan and his students, and subsequently thousands and thousands of people across the globe. And so initially the Drosophila was used for understanding the patterns of inheritance of very different features, morphologically identifiable features under a microscope, basically, or even a magnifying glass, but not so much to understand development. And at some point when they were looking for what is a gene and what does a gene do, and they found out that there is a way to introduce what's called new variations or mutations. Normally it's used, and these mutations can occur in the population spontaneously, and one can study the patterns of inheritance, and the presence and absence of a gene function can tell a lot about how very different features are manifested in an organism, right? For example, when the first mutation that was discovered in Drosophila was called white-eye mutation, and white-eye mutation is because the gene which is normally required for making the red eye in Drosophila was not functional. So basically they called this particular gene required for red eye pigment, but in its absence it will make white eye, right? And this understanding came much later. They didn't know what is the way genes function, but at least they could associate a specific inheritable trait or a factor to a specific morphological feature. And they also could relate that to in the chromosomes, which I'll come to that in a minute. In the context of development biology, what is important is certain kinds of mutants they discovered, right? And that basically is the beginning of what's called genetic control or gene programming development. One of them is this. In 1940s, Ed Lewis, when he was a student, he found this particular mutation, a set of mutations in the population of flies that were growing in the lab. And in this, what you could see here is can I have a pointer, please? This one? Yeah, okay. So this is the normal fly, what we call as wild type. That means it's normal in the sense. So this has 3 thoracic segments. First is somewhere here. The large part of the thorax is the second thoracic segment, and there is a small third thoracic segment. And if you look at the appendages, and all dipterans, flies are dipterans, mosquitoes, housefly, and fruit flies, and they have only one pair of wings. That's why called the dipterans. And this is the actual flight appendage, and there are a pair of small bulbous structures called halteres. They are the balancing organs, and they are present on the third thoracic segment. And Ed Lewis, by using genetic combinations of very different mutants of a particular locus in the initially when even they couldn't define what is a gene, they used to call something like perhaps the factors that are governing a particular character is present in a particular region on the chromosome, and that region was called locus, loci for plural. And they found out that if a particular locus is deleted, which they call ultrapythorax. Don't worry why they call it ultrapythorax, UBX in short. And what you get is a 4-winged insect, so that with the duplication of second thoracic segment, absence of third thoracic segment, so here you get 4 wings. But the reverse is also true. If you have a what's called gain of function mutation, that means where UBX is activated in during developing wing, what you get is 4 halters, right? Suggesting that UBX as a gene, all right, and is required to make this halter. It does so by modifying a wing fate, right? And it is sufficient to make halter. So this is the beginning for people to say that such complex features in our body, as complex as how to make a wing or a halter or such structures, can be controlled by single gene, and which can be actually traced and experimentally even can be manipulated, right? So that basically is the beginning of what we call molecular biology or genetic control of development, the whole new field, genetics of development or developmental genetics. Yeah.
You're saying that only one gene was responsible for this?
Yeah. The one gene with the hierarchy of, I'll come to that in a few minutes. Basically, it's basically triggers a series of downstream events, but ultimately somewhere at the top it's a single gene. Here is another example. There are multiple such examples, and in this example, this is what are called antennae. These antennae are the ones which to sense the external world with the help of chemical sensors. And this is a scanning electron. This is the regular microscope. What you see here is a development of pair of legs in the place of antennae, and this happens because gene which is required for leg development are now activated here. So even before all this molecular biology about discovery of DNA and all those things, people had already sort of reasoned that there are genes. Doesn't matter what's the chemical nature of those genes. Those genes are functional. They're present in every part of the body, but they are functional only in specific regions, and their function can be activated or repressed. If they get activated, a specific structure develops. If it is repressed, those structures are not developed. But still, what is not clear is, in this context itself, for example, antenna making genes should be activated here, and leg making genes should be activated where legs are made. But there are 2 things should happen. One is not only that antenna making leg should be activated here, and all other tissue making genes should not be activated there, right? Because only activation alone is not enough. Repression is also required. In fact, at some point, some of you will talk about Turing's work on activator repressor model of development, right? Or pattern formation. In fact, some of these things were predicted pretty early in the history of development biology. Now, post Mendel, post Darwin, around 1970 onwards, with better microscopy, people started studying cells in great detail. There was a separate stream of scientific community which basically studying cells, and some others were studying evolution, some are studying genetics, right? And again, beginning of 20th century, they all sort of came together and tried to sort of connect each one of them to others. And with better microscopy and other observation, they could make out there are 2 different types of cells, prokaryotic and eukaryotic cell. In our context today, we are going to mostly talk about eukaryotic cell. And they also found out that initially, in the second half of 19th century, people used to call cell as a unit of life. And the beginning of 20th century, people already knew that cell itself is a life. There is no need to call it as a unit of life, right? Even many cells in our own body can be cultured outside, or like for example, you take a drop of blood and you see under microscope, neutrophils will be moving around on their own, sort of, it doesn't need to have any instruction from others, suggesting that cell itself is life, right? And by that time, by the beginning of 20th century, we also knew that every organism, multicellular organism, right, is born as a single cell first, and then the whole new organism is born all the features of its parents, right? Suggesting that the complexity that we see in every organism, both internal, external, complex organs and tissues and the way they are connected to each other. This is actually recreated every time an organism is born, and at the same time, it was not easy to comprehend the complexity of this process. Although we can actually, imagine how complex it could be, right? Look at our body, as we know now that we are made up of more than 50 trillion cells. Another 50 or so trillion cells are kept aside as stem cells. And this body, which is made up of 50 trillion functional cells, recreated every time a human being is born, but recreated from a single cell, right? And fidelity is very high. If 7 billion people are there on this earth, and all 7 billion people have all the features to make them a human species, right? A member of a human species, suggesting that this complex phenomena is highly reproducible, right? And it is, again and again, it is recreated, this whole process. So by 50s, we had sufficient understanding, 50s and 60s, of chemistry of life, with series of discoveries post cell biology and combination of cell biology and genetics. People discovered that the DNA is a genetic material which is present in the nucleus. That's why it's called nucleic acid, and DNA has a particular copying mechanism, and that basically maintains the information with transfer with high degree of fidelity, and how information is decoded is all sort of worked out, right? And people also found out that it is true across all forms of life, which also sort of proves again Darwin's original hypothesis that from a common origin of life, you can actually have through a process of divergence, very different forms or have evolved. And this copying mechanism, semi-conservative mechanism, itself is a fantastic discovery that sort of explains so much about life itself, right? And it also talks about how variations are generated. Whenever you talk about genetics, we need to understand how variations are generated. It's because the copying mechanism is not 100% error proof, and if it is 100% error proof, there wouldn't have been life after the very first day of any form of life that originated. Because there is variations in the environment, which is a continuous process, unless there is some genetic variations are introduced in the population, there is no process of adaptation. If there is no process of adaptation, the organism wouldn't survive, right? So there is a mechanism by which variations are generated by mismatch in the base pairing, but there are also multiple other mechanisms by which variations can be generation of variation can be accelerated, but the primary variations are generated through a small error that is still left in the DNA copying mechanism, which is not evolved to become 100% error proof. And now, if you combine everything, what we know about as of, let's say, 21st century, is life is an organized state of chemical reactions. It has well-defined boundary conditions. We just talk about single cell as a living system, right? And most important of all of these things is the spatial organization, right? Within a cell, everything has a certain fixed position, and what one biochemical reaction happening here, another biochemical reaction happening here, the product of this biochemical reaction may become reactant for the other biochemical reaction, and there should be a specific time scale in which all of these things happen. And because any chemical reaction is basically a stochastic reaction, you can increase the rate of reaction by, for example, increasing the concentration or increasing the temperature, so that you can increase the collision rate. By increasing the collision rate, you expect that some reaction will happen, whether it's, let's say, nitrogen, hydrogen mixing and making ammonia, right? But many of them are so reversible reaction. Unless you take out the product, the forward reaction will not happen in the same rate, right? So what happens in living system is the same chemical reaction. It's extremely definitive time scale compared to extremely stochastic processes that happens in nature outside living system, right? Of course, living system is also natural system. Now, this is what is this particular phenomena. It can be extrapolated across all aspects, including development. What happens inside a single cell is spatial organization of very different constitutions, which generates temporal organization, and that basically is most important for something called life, right? Same thing in a multicellular organism like our body, right, made up of 50 trillion cells. There is supposed to be a spatial organization of very different constituents of tissues and organs, and if that is not properly organized and connected to each other, you will not form a human individual body which functions as one single unit, right? For example, if I'm lifting my hand, billions of cells would be going up and down, right, in synchrony. And to maintain all of these things, the most important is every cell should know what is its positional information, what is its function, and that function depends on where it is present and what is its biochemical identity, right? For example, there may be muscles. There are certain common features of all muscles, but there are muscles of different kinds in different positions along the body, and these had to be conferred to those cells. Unlike a single cell, where every time what is inherited is not only DNA, the spatial organization of the cell, right? When our mother cell divides and forms two daughter cells. But what about development? Because you start with one cell and you go up to 50 trillion cells, but one cell to 15 trillion cells, it has to be still maintained as one single living unit, right? So the complexity of development is at a different scale, given similar kind of underpinning of how this has to be organized. Yeah.
You mean to say that life arises when the objects, when the constituents, this is not working. So when the constituents have come to a definite position and they are not moving after that, it seems that only in that case you are defining it to be life.
Whenever the life originated on Earth, the cellular form of life in 3.5 or 3.7 billion years ago, and they basically had this kind of an organization. And this cellular form of life, because of the chemistry of life in terms of genetic material and its ability to generate new variations and adaptation, because that basically is the one which is surviving for the last so many billions of years. It's not that life originates again and again. Life has originated only one, this particular form of life that we all part of.
Right. What I'm trying to say is that perhaps the orientation of all these objects within the cell would have been different, and that would have led to a different. I'm fine. That would have been led to a different form of life.
No. So that depends on what actually you consider as life, right? So if you consider life as something where it's an organized state of some of these chemical reactions, but it has an ability to inherit, make it copies of itself, multiple copies of itself, but with high degree of fidelity of information transfer, but it also can adapt to changing environment. If you consider only this much, right, irrespective of whatever the form of life, you need to have certain organization, right? Because when say chemical reactions have to happen, it has to have a certain organized state. It also has to happen in a time scale, because the environment is continuously changing. It cannot adapt on its own time scale. It has to adapt at a time scale according to environmental changes, right? So, and along all of these things you put together, what we say today's current form of life has these features. Okay, so one last introductory before I get into development is those who do not have a clarity on what is a gene. Now, gene is nothing but a specific length of DNA which has one particular functional unit, and it has information for a specific function, and this function is mediated with the help of a protein, or it could be RNA itself, right? But to make the protein you need an intermediate RNA. But at the same time, it's not about just coding information to make a protein or make an RNA, and transcribe to make the functional RNA, or translate coupled or a sequential another reaction called translation to make the protein, right? But beyond that, what is important in a complex organism, even if it is a unicellular organism, is the proteins are made specific time point. Not all proteins are made all the time. Taking the unicellular organism, it's more than the spatial location of the protein, it is the temporal regulation is more important, right? Because there is a cost to make these proteins. You don't want to make all these proteins all the time. You make only when it is required. Second is quantity of the protein. You don't want to make too less, it will be not efficient. You also can't make too much. It will not only there's a cost to make this protein, energy cost, and at the same time it will interfere with the functions of other proteins, right? Because they're all chemical entities, they can have interference. So a gene in modern day, right, it has information and not only to make a specific protein or a functional RNA, but also it should have information that when to make the protein in time scale, where to make the protein, particularly in multicellular organisms, in the spatial scale, and how much of the protein should be made. All that information is present on the gene itself, right? This is what we want to decode and how the development happens, from a unicellular organism to multicellular organism. Yeah. How are they?
Different from activators of a gene.
Activators of?
A gene.
A gene. Okay, so the what I'm talking about, all so-called cis-L regulatory elements, that means it is a piece of DNA itself. It is the sequence in the DNA has information to where the activator that you mentioned would bind, right? And or a repressor would bind. That information is already there in the DNA. And also there are sometimes the secondary structure or tertiary structure of this particular region will also have a regulatory role to play, right? So all of these things is put together. Of course, there are other proteins which are involved. They are called transcriptional activators or repressors. I'll come to that in a minute. And depending on what binds, when it binds, a gene gets activated or repressed, right? Okay, so again, this part you don't need to explain again. All of you know that, particularly, if you look at the animal development, all of us are born as a single cell embryo. Embryo, we can call, there are multiple terminology we use just for introduction purposes. When you say egg, it is an unfertilized cell, but it has only half of the genome. It's called haploid. When it is fertilized, by a sperm, it becomes diploid, a zygote we call it as. The word embryo is used only when it starts the developmental process. From one cell it becomes 2 cells, 4 cells, 8 cells, and then the organization start. That is called embryo. And in human we usually use fetus, not so much embryo, right? But there is no much difference between a fetus and embryo, right? Fetus is a medical terminology and embryo is more of a zoology one. And so what is that? From 1 cell to 50 trillion cell, if such a human individual is to be born, first thing it has to generate is a positional information, because from 1 cell you get multiple cells, right? You don't expect too much differences among this in the cellular constitution itself, because it's the same human cell. Second is the genome will remain the same, right? That every time a new cell is born or 2 new cells are born, then genome is copied, right? So genome remains the same. So that means although it has initially no particular information of itself other than it is going to become a human baby, but as and when more and more cells are generated, those cells should have some position information. It should become an eye or a ear or a nose or a part of an arm or a leg and so forth. Second is most important, is should generate size and shape which is specific to the species, right? For example, these 2 arms develop exactly the same size and shape, extremely well-regulated processes and proportionate to the body size according to the species. When my whole body is small, of course these would also be small, right? If I'm very big, and then it'll also be larger, but proportion will remain the same. That's why we can identify each other as human beings, right? And that information should also be generated during development. Then finally, to make a one living system, you should have all the physiological connection, right? So that the brain can control certain kinds of ones. Or for example, blood circulation should happen and supply oxygen to all over the body, and nutrition, or function of the lung and so forth. And many tissues are also interconnected, right? Liver is interconnected to something else, and for example, depending on the glucose levels, the pancreas should get activated and the insulin should be produced and so forth, right? So this is how we need to look at what is development is, right? Now, given that it is the same cell which is making different types of cells, one extrapolation you can have is basically same cellular mechanism, there is an improvised one. The cell has a membrane, plasma membrane. Inside the plasma membrane it has, so it's called cytoskeleton. Cytoskeleton is made up of two kinds of major proteins, microtubules and actin, right? And those are all the same every cell. From 1 trillion to 50 trillion, every subtypes of cells, it will be the same. So, but it may be used slightly differently. Muscles has so much of mechanical force generates. It's, the kind of cytoskeleton will be different than a very soft, epithelial tissue or soft, let's say red blood cell and so forth, right? So obviously, although it's a similar kind of cellular mechanisms, maybe it is used differently in different parts of the cells. And most important in this kind is complex regulatory circuits of cell division, right? A cell, when it divides, whether yeast or other single cell organisms, they follow certain kinds of stepwise cell division process. We call it as cell cycle phases, right? Something called G1, G2, S and M phase. I'm sure many of you know about it. G1 is more like a preparatory stage. Make sure that everything is fine and DNA can be copied. Then it gets into what's called S phase, is called synthesis phase. DNA is copied. Then it goes to a G2 phase and make sure that DNA copying is fine, genome is integrity is well maintained, and all other features and in terms of energy and in terms of very different organelles is all well in place, so that cell can make into two copies of itself and divide. Now, that simple cell cycle regulation is not enough if you are talking about determining fixed size and shape of a large organ made up of billions of cells, right? So you need much more complex regulatory circuits. So this itself can be studied in isolation, right? It's one of the most complex phenomena, and it's also amenable to all kinds of mathematical and quantitative methods, and many people have worked on regulation of cell division. Now finally, because it is the same genome from the same cell, and sort of making this kind of an organism of multiple different subtypes of cells, right? You can sort of reductionist approach is to bring down to studying spatiotemporal gene regulation, right? It's not going to answer everything, otherwise we wouldn't have been here, right? So the beginning of understanding development is spatiotemporal regulation of gene expression, right? But still, then you need to understand this and also understand the cellular mechanisms. Okay. How much time I have? Another 45 minutes. Okay. I'll try to speak for about 30 minutes and then finish. Okay. So again, it's a very introductory lecture. I'll not get into too much of details. Here is a field, and any field in all your graph books and your schools and everything, when you're talking about, or when you want to map a specific area, like map this particular room, what you do is you draw two coordinates, and then give sort of unique values to every point, every spot in that particular field, and then you will have, each one has its own unique identity, right? For this to happen, most important is these two are supposed to be what you call as axes, or you can call it as coordinates, or x and y coordinates, or x and y axis. And the most important aspect of axis is the point, at every point you know the value of this, because they are the referral lines. Along the y-axis or x-axis, you know the values, and using this as a reference, you can assign values to any point, but each of that would be unique. The two reasons why they are unique, one is of course you have two values, not just using one value. The second is they are perpendicular to each other, so because of that you can actually assign unique values with just two values, right? Unique identity with the help of two values. If it is not perpendicular to each other, then you have to add angle between the two coordinates or two axes, then it will have three values to give unique identity, right? Now, during development, if positional information is assigned to, if for example you divide this structure into 4 quadrants with the help of two axes, you can artificially assign values, right? So anyway, in any of these axes, the value that is known is basically you are given the values. It could be meter, it could be kilograms, it could be grams, or it could be any value that you want to sort of is part of your measurements, right? And with 0 to 1 or 0.1 to 0.1 to 0.2 or 0.01 to 1.1, it's up to you, right? But along this path, every point has known value, right? Now, in case of positional information, when we generate, you can also assign the same way, right? Without getting into the details of how it was discovered, at some point, in 1950s, even much before molecular biology was used to understand development, people sort of predicted that there could be a process by which these positional information to every cell was assigned through a process in which there's a values along the line, or we can call it as axis, in the two axes, maybe used some process, right? They call it as some morphogen gradient, right? Some gradient of some information runs along this axis and along this axis, and that basically gives unique identity to every cell in the field, right? And being an experimental biologist, I'll just try and explain with an experimental approach to this. Although initial thought about morphogen gradient, that there is some kind of a gradient information which is generated, and that gradient information is sort of extrapolated to give positional identity to cells, was postulated in 50s and 60s. Okay, so Drosophila embryo is one of the best studied for this purpose, as Drosophila and Xenopus, the two, one invertebrate and one vertebrate model. Mechanistically, there are some differences because the way their embryogenesis happens, the way fertilization happens, and then what happens of the first cell division and so forth, during the first few cell division. But philosophically, at the conceptual level, it's very similar mechanism, right? So the way this is the just fertilized embryo called zygote. At some point, after a certain number of cell divisions, it has very different features, although morphologically under microscope you cannot see, but it has already some positions are already assigned. So what people had done is people actually use certain approaches to kill a small piece of a certain number of cells here or here or here, and depending on what which part of the body had defects, they could assign that perhaps head is coming out of this region, thorax from here, abdomen from here, and so forth. So they call this as a blueprint, right? The blueprint itself is not the organism. Blueprint is basically a map through which very different positions are assigned. But later during development, very different features of a larva develops, right? And now the question is there a way, even without looking at the morphological features, trying to see how this blueprint is made. The fact that blueprint is made, it can be established experimentally by these ablation experiments. Okay, now first aspect of starting to understand what is position and how positional information is generated and assigned to different cells in a developing embryo, you want to know, is there any understanding of there differences between the very different ends of the embryo, right? And if you look at an egg, this is the unfertilized part. You can call it egg or oocyte, technical term. And in a oocyte, there is no, called polarity. So the two ends along this axis, the long axis, if you look at it, the two ends, they are very similar, right? And although they are similar, if you look at how they are positioned in the mother's body, how it is positioned in the mother's body, right? And this is the anterior of the mother, that is the head of the mother on this side, and this is the anterior, and this you can call it as posterior. So you can say perhaps the oocyte also has these two ends known. This is the anterior end of the oocyte and this is the posterior end. Perhaps yes, but then does oocyte know? Then what does it mean by oocyte knowing? That means there must be some molecular features in the egg to say this is anterior and this is posterior, right? Now, if you look at the microtubular organization in the oocyte, you know what's a microtubule, right? It's basically a polymer of tubulin proteins, and there is what's called growing end, and there is the starting one. So the minus end is the one, and the newer subunits are added, that becomes the positive end. So if you look at it, it has a radial sort of a symmetry, right? And there is really no, this is of course a cross section, but it basically is a radial symmetry. So somewhere in the center of the oocyte, it is plus end. So all the microtubules are basically growing towards the center, and there really no polarity along this axis. So again, in development, although we say that from a single cell which is laid outside, even in a mammalian kind of a system where the embryo develops within the mother's uterus, it's basically a single cell zygote is released to the uterus and it develops inside, right? But there is no direct positional information or development related information is provided by mother again and again. What mother provides is the space and the nutrition, right? The uterus provides the space and nutrition. Now, in case of in Drosophila, what happens is when this positional information it is generated, it is basically transfer of already pre-existing position information to the oocyte, because once the oocyte goes, and basically it is a fertilized egg is laid outside and then it goes outside the mother's body, right? And the way it happens here is these are called nurse cells. These nurse cells are very large cells which make yolk to the developing egg or embryo later. As you know, this is oviparous, right? It has to grow outside the mother's body. There's no another source of nutrition. Nutrition is already provided along with the embryo at the time of laying out, and that is called yolk. And that yolk is made in large quantity. And because the yolk is made large quantity, these cells become larger and larger in size. And they'll become larger and larger in size. They push this one oocyte on this side towards the edge of the, this whole ovary, right? And these are all mother's, somatic cells, right? And when they get pushed, there is basically a direct, contact between cells, and that sort of triggers some chemical reactions or biochemical reactions. And that biochemical reaction leads to reorganization of the microtubular structures in the oocyte, and there now it has a clear polarity. The minus end on one side and the plus end on the other side, right? So as you can see here, how positional information can be generated. Here it's not really positional information in a specific way, just to create that two ends are different from within a longitudinal axis of the embryo. Now these nurse cells also produce certain kinds of transcripts. Those transcripts are nothing but the mRNA, which is the product of transcription from the gene, and those transcripts are deposited in the oocyte, right? And if you look at two such transcripts, one of them is sort of get localized to the minus end of the microtubule, other gets localized to the plus end of the microtubule. Now, if you look at, there is no information in the beginning and the two ends. Now you can say there is information, that is minus and the plus end. This information gets now further extrapolated, that the presence of bacoid mRNA here and the presence of Oscar mRNA on this side. Don't worry about the names, don't worry about what they do and everything. Just the two, as you can see here, information is now generated in a sequential way and becoming more and more complex information, right? Now, this particular mRNA is now localized. This is the actual embryo and directly visualizing the presence of RNA using a particular technique called RNA in situ. But when for whatever the mechanism, there are two mechanisms associated with this. One is microtubule associated mechanism by which it gets localized. There's also a bit of phase separation, molecular crowding happens, which also makes the mRNA get localized to the minus end of the, towards one end of the microtubule. Now you can see here, there's a clear definition of what is this versus what is the other end, right? The bacoid mRNA end versus opposite end. Don't worry about what else is there on the other end. But when the protein is made out of this RNA, protein is made, and interestingly, only there is a temporal regulation here. Although mRNA is deposited in the oocyte, and only after fertilization protein is made. The protein is made through a process called translation in the ribosomes. Those ribosomal translation process is suppressed for some time after the oocyte is matured until the fertilization happens. The reason is we don't want, the way the mechanism evolved is, if by mistake development begins, even before fertilization, it will be an haploid individual, right? And that haploid individual with one copy of genome, we can have all kinds of complications because genome, even in somatic cells, can get insulted and can have some DNA breaks and kind of a thing, leading to a problem. So once again, translation is suppressed both in mammals, in insects, both vertebrates, invertebrates, specifically including human, until the fertilization happens. And fertilization is the one which actually triggers the translation, and that basically sets the whole development program on its path. So that protein is made. Yeah, you had a question?
Will organization precedes the mRNA deposition, or it happens simultaneously?
No, sequentially. First time post microtubular organization, then mRNA position. Now, the protein is free to diffuse. There is no, some of the features of the mRNA localization are not associated with the protein. Yeah.
Hello?
Say?
Right. So you showed a diagram where there was these nerve cells which were acting as the yolk. I thought they would provide the nutrition to the the oocyte.
And also some mRNA. Only very few, 1 or 2 mRNA molecules also.
So oocyte grows as a result. Is that right? So
oocyte is not growing in size. Oocyte is basically pushed towards the other end.
Some sort of pressure gradient.
Pressure. It's simple physical phenomena, right? As the nerve cells are growing in size to accommodate production of more yolk proteins, the oocyte gets pushed physically one. The reason that happens, why this is important is many of the, at some point, this workshop has 3 things, right? Flags, signaling, and what is the other one? landscaping, right? So when you talk about signaling, there are multiple kinds of signaling. One is called long distance signaling. A signaling molecule is produced by a cell, released to the extracellular matrix, and it sort of diffuses. And that diffusion rate and everything is very important to establish certain kinds of features during development. In another form is where the two cells had to make physical contact with each other, because the receptor on the receiver and the ligand of the sending cell, both are membrane bound. Is not released to the ligand is not released to external matrix, so they had to make physical contact. Otherwise, there is no signaling happens. The signaling mechanism is called EGF signaling that happens between these follicle cells and the oocyte is through membrane bound receptor ligands. Okay. Yeah.
So after the fertilization, is the diploidy that is what is triggering the protein translation, that is?
Say again, after the fertilization?
After the fertilization. So the translation is happening only after the fertilization.
Yes.
So is the diploidy is something that is triggering that particular translation after that?
It's a good question. So it's not necessarily diploidy. You can actually initiate the translation even in haploid, but only by one or two proteins is one. So the mechanism by which the fertilization happens when the sperm enters, right? And that basically the process itself activate a particular kinase, which is basically through a mechanical force. That kinase reaction is important to initiate the translation, but it is coupled. That's all. It's not that the diploidy induces the translation, triggers the translation. The initiation of translation and formation of diploidy by fertilization of the fusion of the two nuclei are coupled. That's all. Okay, now it's very simple phenomena now. There is single source of protein where the protein is made because RNA is here, but the protein is free to diffuse. Obviously, it sets up a gradient, right? So the positional information along this axis now, without getting into too much complication, it's not just as simple as that there's one protein here and forms a gradient. There are multiple other proteins also operating from the other end for better refinement. But at the conceptual level, you don't need to get into other 3 or 4 different proteins involved in this. So to simplify the phenomena and to explain, it's this. You have a source of mRNA here on one side, but the protein is made and it is free to diffuse. It forms a gradient, and here is your flags, right? So let's say you have multiple cells, but the way after the fertilization, cell division is also happening. The gradient is also being formed. You can call it as multiple cells or multiple nuclei. It doesn't matter. It's the same phenomena, but in different zoological taxa, there may be slightly different detailed mechanism. So all these cells are equivalent in terms of their identities. For your convenience, you may number them as 1, 2, 3, 4, 5, 6, but basically all 6 are the same, right? But now this morphogen gradient information, depending on how much of a particular protein is present, how much is in each cell, you can divide them into 3 different categories. It can be more also, and for better understanding, Louis Wolpert, when he was talking about, he divided them into 3 different categories: blue, white, and red, as in the French flag. And of course, there are 2 things we have to keep in mind, because in these kind of continuous gradient, there is no clear cutoff point to say that up to this is identity 1, then to identity 2 and 3. It's basically a range of the quantity of those proteins, right? You can give some arbitrary number, let's say unit, and 1 to 100. So it's not like up to 10 is one identity and 10 to 20 is another identity. It will be a range, right? You can divide it largely into 1/3, 1/3, 1/3 kind of a thing, right? The highest 1/3, lowest 1/3, median 1/3 and kind of thing. So that are threshold levels, and this is what identities are generated, which is indeed true, right? Now it has been experimentally shown. I'll skip some of these things. I don't have all those slides here. People have experimentally shown what happens if this gradient is not formally formed. You can see that it may form only the head structure, but no other structures are formed, suggesting that this gradient information is not percolating. Or you increase the, put this here also, you'll have two gradients coming from both sides. Then you can see two head structures. Or if you remove this, then you don't see any of the anterior structures, you only see the posterior structures. That's why it's called bicoid, right? So two tails, no heads, right? So you can experimentally show all of these things, that indeed such a gradient is formed and the gradient information is important.
Why is the threshold different in action?
Why?
Is it because there are different enhancers, or like why is the threshold different in action?
Yeah, I'll come to that in a while. But if you look at the what is the function of this gene and how it functions, or right, you see when the proteins are made, what is the amount of the protein that is required for its efficient function, right? And it's always a certain range, because most biological process, there is nothing like very precise amount of protein made at a given time, right? It basically is always a certain range of proteins. It also adds robustness to the system, right? It is not too less or too more, right? In certain range it helps in the robustness. For example, here itself, if you look at the gradient is also a function of the temperature in some extent, right? So amount of protein that is diffusing from one end to other end over at some, after a certain time point, let's say there is certain amount of protein present in every one. Beyond that time point, if protein is still being made, right, then it will get more and more protein gets added. Or if protein synthesis stops but diffusion continues, then less protein is present here and more protein start going in the other direction, right? So how do you interpret the results? What happens certain amount of protein is present? So very different experimental methods and of course computational methods interpret that. As a specific time window is important to interpret the function of these proteins, right? After that time window, the gradient itself is look different. For a given cell, how much of that protein is present will be different in different time points. Second is the rate of diffusion. Let's say it is slightly higher temperature than the normal. Let's say increase the rate of diffusion. That basically may alter the gradient in terms of how much units. So if it is an absolute unit you talk about, right, then it's very difficult to interpret the experimental results. But if you say it's not absolute, it's relative amount, certain range of proteins are present, then most of these experimental results can be interpretable. And also it explains the robustness of the system, reproducibility of development, because it's otherwise they'll be continuously making errors. Maybe 1 in 1,000 would survive, but here the reproducibility is pretty high. Almost, if you give right nutrition and temperature, every embryo will ultimately make a fly.
Am I interpreting this correctly? So you have a certain mRNA on this boundary. This mRNA is producing lots of proteins. I mean, they're being translated and you get lots of proteins, and there's a protein diffusion. And that protein diffusion is leading to different kinds of cells within the organism, where the higher concentration is leading to, say, as I understand,
Head
this
structure.
is the tail. So it's producing tail.
No, tail is the other end, the head structure.
Okay.
Highest amount is the head structure.
So that's going to produce the head. Okay.
This.
If we talk about the relative gradient as well as the absolute concentration, so is the relative gradient of the basically from one point to other more important than the absolute concentration of that particular protein in the different basically? So if we divide the egg into say 6 segments, and the absolute concentration of the protein in those segments.
No, threshold levels are more important. There is certain threshold level is really important. It's not only relative. If it's only relative, then it can start from any level, right? But there is some minimum threshold level is important. Otherwise, it will not function. So the reason is, in bicoid, this particular example that we picked up, It's a transcription factor. It binds to the DNA of other target genes and activate or repress the downstream ones, right? Now, how much of this is present will tell you how well the downstream genes are activated or repressed, right? And some genes have one binding site, some genes have multiple binding sites. So if it's only one binding site, you bind and then it cannot get down regulated. If there are multiple binding sites, whether all of them are bound or partially bound, also will tell you what should be the threshold level that is required, right? And given all of these things, it is the threshold level which is important. There are also additional complexity. There is also another gradient which comes from the posterior side as an inhibitor of this transcription factor. Physically it inhibits, there is a competition for binding sites. And there again, it sort of sets the range and the threshold levels, right? I'm just explaining very simple way because it's for fundamental conceptual understanding. What is important is the formation of a gradient, and that gradient information is extrapolated into positional information.
So if at the anterior position, if we basically, so if we are overexpressing bicoid, and then that is basically diffusing at the same rate as it happens in the wild type, will the tail structures be affected because the threshold, so basically expression of bicoid is slightly above the threshold that should have been in the tailed structures?
Yeah, it happens. I told you already. So there is a gradient of another protein which is coming from here. If that particular protein is absent, or if you express this also on this side, and you'll have more of the head forming protein, and that basically will all disturb the position information. So in a way, it's actually pretty linear in a way. The presence of bicoid protein, the highest amount of bicoid protein means its head structure. It doesn't matter whether the highest amount of bicoid protein here or here, it forms the head structure. And the absence of bicoid protein, that goes to the default state, which is more like a tails kind of a structure. And that's also true. If you remove bicoid, you will get a tail-like structure. And it doesn't have to be, again, remove below threshold level. It doesn't have to be complete removal of bicoid. I mean, you don't need to make it to 0. Okay, very quickly. Now, as you can see here, depending on the amount of the protein, very different genes can get activated. Again, it's too complex for me to explain to this audience because there are multiple other cofactors also come into picture. It simply say that depending on the amount of bicoid protein is made, right? The different downstream genes are activated, right? And there also, this is a sequential hierarchical event. I'll skip all of these things, right? And there are also experimental evidence for this, okay? And basically starting with no information in the embryo, not even polarity, you generated polarity, you generated position information along the axis and the referral. Now you actually see distinct proteins are getting expressed in different domains in the embryo along one axis because of the gradient of these proteins, right? And so this is how the position information. Now you can say this is head, this is going to be the thorax, this is the first part of the thorax, second part of the thorax, this is going to be largely the abdomen, and this is going to be the, there are two parts of the abdomen. There's the first half of the abdomen is mostly for the gut and other digestive system. The second part of the abdomen is mostly for reproductive system. So the abdomen also can have a very clear, distinct two features. Now, as you can see here, this is one of the reasons why I keep saying that there is no really absolute unit here, right? So the boundaries are not very sharp, right? But they are somewhat overlapping gene expression pattern. But with time, with more and more genes get activated, and they are, talking to each other, activating or repressing each other, or sometimes interacting with each other additively, or sometimes they, negatively interact with each other. Over time, what you see is formation of these sharp boundaries. Don't worry about which gene, why I'm showing red and green here in these ones. But what is important here is any gene expression, if you look at as a which gene which confers positional identity, that gene expression has become sharper and sharper now, right? and because we are looking at difference along this axis. So along this axis, everything is identical as of now, because you are not looking at any specific proteins. Whereas along this axis, you can see here that there's red here, green here, some red here, green here, and different green and red here, and right. Finally, the position information is not because of one particular protein; it's a set of proteins, right? These group of cells will going to form thoracic part. These group of cells going to form something else. But later, when initial identity is conferred, although from a single protein, let's say one or two gradients, then the larger, like head versus thorax versus abdomen, or within the thorax, first part of the thorax, second part of the thorax, a group of cells are given identity with the help of group of proteins. But later, when you start giving identity to a specific subset of those tissues, right? Again, sometimes single gene can come into picture. For example, the very first one I showed you, UBX, it is expressed, and it can make halter or it can make wing, depending on present absence of that particular protein. But a group of cells to make wing or halter come because of 3 or 4 different other proteins which function upstream of UBX, right? So as you can see here, the interplay between various different genes, where you're getting activated or repressed, right? Some are cooperative, some are not cooperative, and that basically confer positional identity along the axis. But it all starts with 0 information, and you go on adding more and more information. But why it is highly reproducible? Because the mechanism by which information added is conserved, and also is reproducible. And for example, in Drosophila, right, the first step is position information of the mother is transferred to the egg, which is anterior, which is the posterior, the polarity information. In case of Xenopus embryos, I don't have a picture of that one. Sorry, I sort of underestimated how much time I would take in giving the background information. I just showed this movie once, although it's it's not playing. Okay, so in case of frog, sorry. No, it's actually I think it's not properly embedded here. It's okay. Right? In case of frog, an egg is released by the female, and the sperm is released by the male. But it happens both. Fertilization itself happens outside, right? Again, this also has large amount of yolk present in the egg, right? But it happens in the water, and typically what happens here is the yolk being heavy, which is in the bottom, and it is not completely filled. 2/3 of the volume is yolk, 1/3 is left with other cellular stuff, and because of which the 2/3 of the egg is submerged in water, and 1/3 is above water, and the sperm which sort of released and is swimming on the surface of water, and invariably the interface between the submerged and the outside is where the fertilization happens, because as the sperm entry happens, not fertilization, sperm entry happens, right? And this basically leads to some reorganization here, and that sets up the 2 polarity, right? So the entry of the sperm, that point becomes the future ventral side, and the opposite side becomes the future dorsal side. If you look at in the fly embryo I mentioned, the presence of this polarity okay, again I lost the connection. In fly embryo I mentioned minus end plus end of the microtubule sets the polarity. Then you have some bicoid RNA, and it sets up the gradient. Here again, some gradient of this comes from this side, right? Because of this, the mechanical turbulence that is generated because of the sperm entry will reorganizes the embryo as a oocyte, or now it's embryo or zygote. And that basically sets up some chemical reaction, biochemical reaction, formation of some kind of a gradient of a protein. Gradient is in this direction. Highest amount becomes the dorsal, lowest amounts the ventral. Yeah.
So going back to the fly patterning, so can one sort of decouple the temporal nature of finer and finer borders being set in? Let's say the hairy runt genes which were being expressed for finer boundaries. Once the syncytial divisions have occurred, can we use some other driver to express them before even hunchback nerves they set in? And if we do, what sort of patterns do emerge?
You mean you sort of activate those proteins even before the gradient is
Yes.
established, right? So, it's very simple. People have done this experiment. The fun is, of course, you can actually suppress the formation of gradient itself, right? So, some identities will change. Now, let's say if you, see, as soon as the fertilization happens, right, that's when you start making the zygotic changes. You can't make changes to any of those genes before fertilization. Once the fertilization happens, you make those changes, and you can actually now make the bicoid gradient to express, right? And that happens in the time scale, which is, let's say, time t is equal to, let's say, 1, right? And time t is equal to 2 is when other genes get expressed,
Yeah.
right? Because of bicoid. Now, what you are trying to say is, can you activate those genes right here, even
Yeah,
before
cap
bicoid?
genes before bicoid expresses.
Yeah, such experiment has not been tried. For main reason is, experimentally it's difficult to activate those genes, at the same time when the fertilization is happening, right? So one possibility is, let me get, I don't know if I'm an experimenter. There is the modern way of making it. There is for example, you could take a universal promoter, which is expressed anytime, all the time, everywhere, and then you activate this, keep this, that means this protein is expressed all the time, right? So as soon as it is expressed in the oocyte itself, it is also expressed in the zygote. So that means, what happens to that, we need to find out. I don't know if people have done this experiment, I can check out. My guess is it will definitely be lethal. Let's see whether it will have some significance in terms of precocious positional information. Say it again. I don't know why is it not. Yeah. Oof. This one?
So my doubt is, so maternal bicoid mRNA, we just see that it just declines in the start, but yet there is a gradient of bicoid protein. So how is that? Shouldn't it also
Yeah, yeah.
follow the same pattern or gradient?
No, I didn't understand. So this is only it is not temporal aspect, it's only spatial information, right? Temporal also, for example, bicoid is not made in the fertilized egg. It is only maternal. So there is a finite amount of bicoid mRNA is present, and that get translated, right? and you make finite amount of protein. That finite amount of protein will form the gradient, which now spatial information is generated, right? Temporally, there is only one information, that is maternal versus zygotic. The mRNA is maternal and the protein is zygotic. The reason it is shown here is it just occupies so much of space in the anterior end. You cannot simply say that it is right here, only this much. The mRNA itself occupies, if you look at experimentally.
So after its translation,
No, mRNA is, this is mRNA, this is RNA in situ. It's only visualizing mRNA directly, right? This is mRNA and this is the protein. You can see here protein is going beyond the space where mRNA is present.
So after the translation, it will follow the gradient, not
Exactly. Protein is the one which is forming the gradient, not mRNA. mRNA has a fixed location and protein has. Okay, I'll skip all of these things just to say that the experimental proof for every one of these things. What is the positional information, which gene is involved and which gene is get activated when, and all those things up to the positional range. Finally, what I was trying to say is, with almost no information in the egg, but the information is generated afresh in that particular generation, which is forming a new individual, with the help of information which is provided through inheritance, right? Genetically, you generate a gradient of this protein, and downstream of that, certain genes get activated, right, in different parts of the embryo. These genes further activate or repress other genes, and because in this sequence of events, there is more information there is more players to generate position information, and this makes the boundaries more sharp. And all of these things precede the molecular events. Under microscope, when you see is, even at this stage, you hardly see any morphological features along the embryo axis. But if you start looking at this, now it will show these things. So depending on which protein is not properly expressed, if it is mutation or because of some problem with the gradient, you will see variations along the body structures, right? Okay, so what we have conferred is basically positional information saying that each one is somewhat different, but we still not make them completely different in terms of development, right? So for example, if I remove UBX, what you get is 2 times second thoracic segment, 4 wings, right? There is duplication of information, but there is no separation of 2 different thoracic segment. So that information is generated in the 4th phase of the hierarchical event. So phase 1 is this gradient, phase 2 is the first targets of the gradient protein, and the 3rd is the targets of these ones. And the 4th is a set of protein which are expressed in different parts of the embryo as a product of these 3 steps, not independently as a product. It's a hierarchical, dependent, it has to go in a particular sequence. Then these proteins are expressed, and depending on which protein is expressed, this is basically the reason sometimes we show larva, sometimes adult is, in Drosophila, the life cycle, what comes out after embryonic development is a free moving larva, which is a feeding stage. Then it metamorphoses and then gives rise to adult structure. But the same events also determine both larval and adult structures. And here is basically talking about larval structures, head, first thorax, second, third, abdominal segments, and so forth. And depending on which protein is expressed, you will see different features coming in. Those proteins are called Hox proteins. Something which UBX I mentioned is a member of those Hox proteins, this one. So you do have, without getting into too much of complication, this is experimentally and in terms of detailed mechanism, molecular level, is also a very complex mechanism. Along the other axis, what I was showing is the long axis of the embryo. Along the short axis, also there's a gradient information of another gradient. Finally, you need 2 axis, X and Y, to give unique identity to every cell in the field, right? Only one value is not enough, right? So that's what, right? For example, all the cells along this line, right, will have the same identity. The difference will be here between this and this, because the axis information is here. So if you have one more axis which runs along this perpendicular to this, then you will have two axes, then every cell can have clear identity. The only thing is, it will create left-right perfect symmetrical body, which is true. Most organisms, including us, are more or less left-right symmetrical along the left-right axis. There's a small difference in the left-right axis, like for example heart versus liver and kind of thing in vertebrates. It's not so much in invertebrates, and that is generated through a third axis, but third axis is generated much later, but it has a minimal role to play during development. The major role is played by the first two axes. One is called anterior posterior axis, another is called dorsoventral axis. This is ventral for us, and this is dorsal. This is anterior, and the other end is the posterior. And how the information generated, I'll not get into the details, but basically conceptually it's the same. There are certain protein gradient is formed, but molecular mechanism very different, and that protein gradient runs along this axis. So if you take an embryo which has a hollow in between, the cells are all on the periphery, right? And if you look at this is the one, and here is that particular protein expressed at highest level, and when you go like this, it sort of tapers, right? So this is your dorsoventral axis, right? This is the one which becomes the future ventral in case of Drosophila, and the other side becomes the future dorsal, right? Also, it's interesting, there are very different evolutionary aspects one can look at it in this case. For example, the primary axis, the way it is generated in case of Drosophila, is the anterior posterior first and then dorsoventral. In case of vertebrates, it is the dorsoventral first and then anterior posterior. And also the molecule which is required to make ventral side in Drosophila is the similar molecule which makes dorsal one in case of vertebrates. So there is some shift in what is primary, what is secondary, and there is some inversion also has happened, which also can be explained looking at the molecular mechanism, right? For example, in case of Drosophila, the nervous system is on the ventral side. In case of vertebrates, the nervous system is inside the dorsal tube on the dorsal side, right? And what's called notochord. And that inversion in evolutionary time that has happened, and one can explain looking at this molecule, because molecular mechanism has not changed, is more or less the same. What confers the identity in the beginning. It's just that the nervous system is on the ventral side in Drosophila. The same gene would be involved in the specification of where nervous system should be positioned in vertebrates, but although it is on the opposite side. Okay, I think okay, so maybe 5 minutes. I'll just pose some questions. We'll come back again. There is still, what we are experimental people will sort of have come out with some ways to learn information. Perhaps it's still too simplistic one, because diffusion itself is not so well worked out. As there has been much better refined mechanism now worked out in the last 15-20 years, but still there is more to one, right? Because in vertebrate there is a very different way diffusion happens, because the diffusion happens along the plasma membrane too, whereas in case of Drosophila, its initial diffusion is only through the cytoplasm. Reliability still need to be worked out. What is threshold limit means, and why, what is the range in which it can function efficiently, how robustness is created, and kind of thing. At the same time, there's also very high precision. Precision is, you can millions and millions of flies which are generated, have exactly the same features and size and shape and everything. And there are also multiple other developmental mechanisms which are involved. Beyond morphogen gradient, there is also a whole field called self-organizing system, through which, see, for example, what you are now using the morphogen gradient give broader position information to make different groups of cells, right? But at the same time, within that, for example, where eye is made, right? Or which, for example, in the ectoderm, which becomes the neuronal stem cell versus which becomes the epidermal cell. These are all organized within themselves. There is no external information coming here. There is a little bit of competition among themselves will set up self-organizing system. At some point, you're going to study self-organizing system in dictyostelium in this workshop. Similar kind of mechanism operate, but completely using different signaling molecules compared to what dictys uses. There's also induction. Many times what happens is a group of cells will take up certain identity and they induce other cells in the periphery to take up the same identity. This is a process of induction. It happens during Drosophila wing development. We can show that wing identities of certain kind is through a process of induction. Lateral inhibition and self-organizing system are more or less similar. In lateral inhibition, you take one identity, but you inhibit other cells to take up the same identity so that there is a better pattern formation around that cell. Recruitment in induction are somewhat similar. Induction and recruitment, why they are kept separately is, in induction, a large group of cells are make to take up that particular identity. Or let's say you set up a control cell division one. So some cells start dividing and then they all make thousands other cells to divide at the same time. So there is synchrony in cell division. In case of recruitment, identity itself is conferred to the neighboring cells by one cells by recruiting one at a time. So coming to what I mentioned, that we can sort of reduce the whole problem into spatiotemporal gene regulation. So activating different sets of gene in different cells is finally what development is all about. But is it true? Because finally, you can have let's say different shape of cells, right? And those shape of cells are conferred because of, let's say, gene regulation, and some are long columnar ones, some are cuboidal, some are very spherical, and so forth, right? And when they come together, they occupy a particular space, and also, let's say there are thousands cells of a one particular type, 500 other type. When they come together, they make only certain kinds of shape. They cannot make any different kinds of shape. So there is a definitive way shapes are determined, and that's also important for organism function. We can't have arms of any shape. Only it has to have specific shape. Fingers cannot have any kind of shapes, right? So given all of these things, how shapes are determined, how much of that is directly because of which gene is expressed, and also how much is because of the event that is downstream of it, which is not necessarily directly related to protein functions. So basically there can be lots of forces that are operating. You just, let's say you make larger cell, certain kinds of molecules are expressed, then certain forces operate, right? Also genetically controlled, but then forces will take over the role, not necessarily biochemical reactions. So we can discuss some of these things, and I'm sure during the workshop many people will discuss many of these points, and I'll also discuss one out of these things later, right? So I'm going to discuss these part through some of our own work, recent work on cell shape and size, how it determines the organ size and shape, and how the forces are involved in this, and how only using the forces you can determine the shape and size of an organ, although those forces are the product of some protein function earlier. But other people will talk about very different other aspects of developmental mechanisms. At some point, if time permits, and I'll also can talk about evolution of what called evo-devo, evolutionary development biology. For example, very different forms of life, how they have evolved over time, right? For example, snakes have a particular body shape, right? and how they have evolved, and because the shape of the body also determines their life history traits, their behavioral one. Right? Snakes crawl on the ground, and for example, even if they have some rudimentary legs, why their legs have become rudimentary. Some of these things also we can nowadays ask questions and try to give satisfactory answers. Sorry, it took longer. Thank you. But I'm happy there are lots of questions in between, so I didn't.
So as there were quite a few questions during the talk, maybe we will break for coffee now. So coffee is just outside the auditorium, and please try to be back within. Okay, so we'll maybe give you 10 extra minutes. Is that okay, Shakti? Yeah. So maybe at 12:10 we should be back here. Okay. So coffee and tea are just outside. And if any of you haven't already registered, just get in touch with Nandini. Can you just wave your hands? So please contact